So I'm going to speak about homing endonucleases, which are also known as meganucleases today. So till now, you have heard a lot about the endonucleases. So what are the endonucleases? If you can just write a two-word answer, what are endonucleases, or overall what are nucleases? Endonucleases will cut DNA, cut anywhere in DNA. Okay, will they cut DNA from the ends? Both the ends will endonuclease cut. Endonuclease will cut. Okay, no. So the endonuclease will cut DNA from where? They may cut, yes, in between. They may cut DNA anywhere, correct? But not on the ends of the DNA, yeah, but any intervening site. So we are going to discuss about some special type of endonuclease today, which we call as meganucleases. So let me go to the slide which I presented in the last lecture, the central dogma, and you know about DNA being transcribed into RNA and getting translated into a protein. So this was discussed in detail in the last class. So this is a 1993 Nobel Prize in Physiology press release. It was given to researcher Roberts and Philip Cesar for their discovery of split genes. So many of you may be already aware about what are split genes. If you are not aware, those who are new to this subject, so let me tell you that the genes of lower organisms like bacteria are continuous pieces of information which produces protein by the processes of transcription, which produce a messenger RNA, and subsequently translation, which produce a protein as per the central dogma, which I showed you in the earlier slide and discussed in the earlier class. But in higher organisms like humans, the stretch of information that produces protein is not continuous. Here, the information is disrupted. That's why the intervening informations are processed after transcription takes place. And that process is known as RNA splicing, which gives the actual messenger RNA, and that goes on to produce the protein. So the higher organisms are having split genes. Their genes are not continuous, and you can see this simple picture of the sequence of events radically changed through the discovery made in 1977 by Roberts and Sharp. They found an individual gene can comprise not only one but several segments separated by irrelevant DNA. So this word I would like to focus here. This is the press release from the Nobel Prize in Physiology and Medicine 1993. Is available in the website. So you can see the split genes have some sequences in between them which are irrelevant, which have no meaning in a way. So these continuous genes exist in organisms more complex than those studied earlier. Okay. Now, due to the split nature of the genes, there are a lot of medical implications. Certain hereditary diseases are happening because of these. And overall, the estimated number of hereditary diseases at that point of time was around 5,000. That number is changing because we have now many other informations available like SNPs and so on. Now, these type of genetic diseases are due to faulty protein, which forms part of hemoglobin in red cells. Particularly, we are referring to here One example, the beta thalassemia. The protein is called beta globin. So you can see this picture of the normal beta globin gene and the beta thalassemia gene. Because of inclusion of some of these irrelevant information or some kind of splicing, the globin gene is not getting expressed in a normal way, and the disease is happening due to this. So here they're telling, if no or badly functioning beta globulin is made, the lifespan of the red blood cells is shortened, which results in anemia. In different patients, small defects in the genetic material have been found, resulting in errors in the splicing process and thus in the synthesis of poorly functioning beta globin. In the upper part of the figure 3, you can see the normal. If the globin gene is damaged, which is being marked here with the arrow below, it may, for example, lead to the formation of a larger than normal exon during splicing, or to the formation of a completely new exon. So all these defect or defective splicing causes beta thalassemia. So that's the medical implication of their research, and for this they were awarded the Nobel Prize in physiology. So now going back to this earlier figure we discussed, we know that DNA gets transcribed to mRNA, and there are certain post-transcriptional modifications, right, where these intervening regions or the irrelevant regions, we call them as introns, and the genetic information shown in red here goes to the protein, the circular figure you can see below, and the intervening region are removed altogether. So this is a normal process of a cell producing a protein from a split gene. Okay, so this is known to many of you, more or less. So that DNA is irrelevant DNA, right? Now, there was a term called junk DNA. There is a term called junk DNA, and this was popularized by Susumu Ono for segments of DNA that have no known function. So that intron is kind of a junk DNA, okay? And the statistics is quite alarming, including introns and other such sequences. Our genome has more than 98.5% junk, it is said, and only 1.5% has some meaning from the context of what was taught earlier. But today we know that that junk is not that irrelevant. So anyway, Ono is known for his work on the gene duplication, and he actually reported this for the first time, and he popularized this term as the junk DNA. So as I was telling you, this junk DNA is not that irrelevant or meaningless. Today we know it plays many roles inside the cell. So some of the direct roles that intron sequences play is they regulate the alternative splicing, they enhance gene expression, they control mRNA transport or chromatin assembly, and introns in the 5' and 3' untranslated regions affects nonsense mediated decay. So these are some of the direct roles these junk DNA or the introns or the irrelevant DNA plays. And they also play certain indirect roles, like different ordinal position of introns within gene may have a different functional role. Introns length matters in the efficiency of natural selection, so it plays a lot of role in evolution. Introns can provide a source of new genes. They're meaningless, it was taught, but they are the sources of new genes. So in future, if some new genes are going to emerge in humans or any other organism, it will emerge from these introns. So another indirect role they play is trait-associated SNPs are enriched in introns. Introns harbor several kinds of non-coding functional rRNA genes, and some of the non-coding genes, RNA's importance we discussed while discussing the nucleic acid binding proteins, how they help RNA binding proteins and DNA binding proteins work together cooperatively. Okay, now in the normal process in this figure, you can see the introns are thrown away or rejected or dumped, and they are not carried to the protein molecule. You can see only the red strands, red information coming to the circular globule over here in the below, and this light color introns information is not available over there. But there are exceptions where these introns are not thrown away, they are actually retained, and these mechanisms which retain them are known as intron retention mechanisms. So here different isoforms can be produced from a single gene through alternative splicing. So you see these blue colored blocks. These blue colored blocks are basically the exons, and these numbered blocks 1 to 3, they are the introns. So you can see immediately in the next picture the 3 consecutive exons has been retained, and in that process they have retained one the exons are retained as usual, and now you can see that block 3, number 3 intron has been retained in the transcription stage. Okay, now there are different pathways which follows this. So if you go to case B, you will find that the IRIs are degraded by the nonsense mediated decay NMD pathway. The reason being that retained introns often contain premature termination codons that trigger NMD with intron retention. So the first scenario is this: if they include the intron number 3 over here, or 1 or 2, whatever, so the process won't go further. It won't go to the translation because there may be some kind of termination codons located here, and as a result, this will undergo the process of decay. So although the intron was copied in transcription, it could not go further. The pre-mRNA got decayed. The next scenario is that these IRIs are retained in the nucleus, and in response to stimuli, these IRIs can undergo further splicing to remove the retained intron before being exported out of nucleus for translation. Okay, then it becomes a normal mRNA construct, and it will be translated. In case D, you have cytoplasmic splicing. Okay, the construct will come to the cytoplasm where it will be spliced. The IRIs are shuttled to the cytoplasm for preservation and may be subjected to further splicing with intron retention. And in number D, what will happen? IRIs escape from the nonsense mediated decay pathway and are translated into protein isoforms. So these will be a new form of a protein which is having a intron, and this intron did not have a termination codon, so it will get translated into protein isoforms, which compared with normal protein isoforms are often truncated and may lose domains. However, it could also be that the alternative protein isoforms include extra domains formed by the amino acid sequences translated from retained intron. So you have a lot of possibilities of here. Some of the exons may get dropped in the process, or you may have not just one intron included in the protein. You may have 2 introns included in the protein. So that's why you can create, nature creates a lot of diversity of these protein molecules. Now there is another kind of technique. So you here in this case E, the intron actually escaped the NMD pathway, and it finally made it to the protein level. It is now part of the protein expressed, right? So RNA splicing could not remove the intervening sequence, and it landed up in the protein. But that doesn't mean that it really has a safe escape. The protein itself also can get spliced, and this process is known as protein splicing. So another concept you need to learn at this stage is exon versus intron are the sequences which are in the gene level or in the mRNA level, while extine versus intine are the corresponding exon and intron sequences, but they are referred to as extine or intine when they'll end up in the protein sequences, right? And then in protein splicing, if you can see this blue figure on the left side, the intines are sometimes removed, okay? Because this intine was originally not intended to be part of the protein molecule. So there's a removal of this intine molecule. So this intine splicing involves no accessory enzymes. The intine segment catalyzes its own release as a free polypeptide. So these are some of the fundamental concepts that you need to know. The concept of split genes, where there are introns, and these introns are irrelevant. They are thrown away in the processing. But some of the introns make it to the protein level. But sometimes, even after making up to the protein level, they may be removed from the protein molecule by the process of protein splicing. And the removal at the RNA level is known as the RNA splicing. And these intines or extines, as you can see here, the word intine derived from intervening proteins. They are protein sequences that catalyze their own removal from a longer protein, splicing together the two surrounding protein halves in the process. And this was first discovered to be encoded within the VMA gene of Saccharomyces cerevisiae that encodes an ATPase protein. Okay, these are very, very important papers. You can see over here, and you can read the original copies. Okay. Now, these introns are not of one types. They may be of various types. They may fall into the group 1, group 2, or nuclear pre-mRNA introns, or they may be transfer RNA introns. These group 1 are found in some RNA genes and splices itself out of genes. Group 1 introns fold into a type of secondary structure that has a 9-loop stem that is required in order to be spliced. This group 2 is actually wrongly labeled here, found in mitochondria and chloroplast. They are self-splicing, but they cut themselves out differently than group 1 introns. They too fall into a secondary structure like group 1 intron. So that arrow is supposed to be from group 2, not from group 1. But their splicing produces a lariat structure. A lariat structure is formed when an intron folds back on itself after an exon is cut from it. The nuclear pre-mRNA introns are found in the nucleus in protein encoding genes. Their removal requires the presence of snRNAs and several other proteins. And the transfer RNA introns are found in tRNA genes and needs enzymes in order to be spliced out of the genes. So these are the various types of introns, and this is the information where they are found. Now, if you look into the distribution of the introns, you can see here that introns are commonly found in the empty genes which are conserved at the fungal mitochondrial genomes. The genes here involved in the ATP production, like genes of ATP synthase subunits, then oxidative phosphorylation, like genes of NADH, then apocytochrome B, cytochrome C oxidase subunits, or they may be in the mitochondrial ribosomes, like genes of large and small RT-RNA subunits. The occurrence of introns hosting G, I, Y, or LD genes, the commonly found empty genes are being shown here. So we will discuss about these G, I, Y, and LD genes at a later stage. So, but in this study, they found out that 50% of the introns were located in the COX-1 gene, and 80% are group 1 B introns. Now let us go to some interesting observation in the early 1970s, which scientists came across at the Institute of Pasteur Institute in Paris. Here they found some kind of deviation there that the principles of genetic inheritance described by Mendel were being violated when they were performing certain process with the yeast strains. They discovered that a particular mitochondrial allele, which later on came to be known as omega, was unidirectionally inherited, and this was later determined to be the gene for the large ribosomal RNA subunit. Okay, that was one observation by that group in the Pasteur Institute. Now, later on in 1970, Boss and his co-workers published a paper in Nature where they reported visualizing an intervening sequence within a yeast mitochondrial ribosomal RNA DNA with the process technique of electron microscopy. This is one of the very, very interesting studies. You can image DNA with electron microscopy, and they found out this particular intervening sequence in the yeast mitochondrial genome. 1 year after that, Fay et al. Described that sequence and organization of this yeast element, concluding that the rDNA was interrupted by an insertion of approximately 1 kilobase. Okay, so earlier guys, they imaged it, and later on Fay et al. Actually sequenced it, and they found out, they described this sequence and found out that it is actually containing some kind of a insertion. There is some kind of interaction in this particular rDNA gene. Together, these basic work provided the initial details corresponding to a locus in the yeast mitochondrial genome, termed as omega, which was not following the Mendelian inheritance. Okay, and this particular construct or this gene later on was described through the process of homing. So what is homing? We are going to discuss. I'm just trying to build up the conceptual framework step by step from where all these came, from which are the seminal research papers from which all these concepts has been developed. So now in 1980, Duzon determined the complete nucleotide sequence of that mobile element, which was earlier observed through electron microscopy, later on described by Fay et al. That it was having an intervening sequence. So Duzon actually sequenced it, and these actually fell into group 1 intron, that type of introns we were discussing in the first place. And it is did for several yeast strains. The intron was found to be exceptionally long, 1,143 base pairs, and contain an apparent reading frame that might encode a 235 residue protein. Now, in the beginning, I showed you that split gene discovery, for which the discoverers were awarded the Nobel Prize, and we refer to that DNA as irrelevant. And in between, I was telling you, introns are fragments, intervening fragments, which are discarded in the process of mRNA, pre-mRNA processing. They are discarded and they never make it to the protein. Then I told you, sometimes some of these introns are lucky enough to be included in the final transcript, but it doesn't move further because that particular transcript containing the intron will be, destroyed by the cellular mechanism. In spite of that, some of these introns may make it to the protein, but even then, it won't go further because at the protein level also there is a phenomenon called protein splicing, whereby the product of that particular intron will be discarded. But in spite of all these, there will be very few, not few actually, substantially quite a large numbers of introns. They will make it to the final protein product. Anyway, these introns are not that big sequences. These introns are very, very small sequences. So it was actually a kind of a, revolutionary idea when Dusan determined these sequences, and he predicted that these introns are not small introns, they are very big introns. And within these introns, you have a likelihood of having a sequence for a full protein, not a fragment of a small protein. Okay, it could be a potentially fully functional protein, and it together, if it is coded, it may be around 235 residues protein. So this was something unknown of at the time. So this slide we have more or less discussed about the introns being purely junk, but later on, now with Dusan's discovery, we know they may not be junk. Okay, so the behavior of the omega intron was because of this particular gene, which was found out by the work of Dujon et al. And it was later on found out that this particular gene within that intron encodes a DNA endonuclease that recognizes and introduces a double-strand break in the LSU rRNA genes lacking the intron. Okay, so that particular gene, which may be around 235 amino acid residue long, is basically endonuclease. And then this endonuclease is something very, very unique in its entire character. So using these endonuclease, this important work came out. SCE is Saccharomyces cerevisiae. SCE1 is Saccharomyces cerevisiae 1. So this is some kind of a endonuclease which is used for inducing a homologous recombination in mammalian chromosomes. So both these papers are seminal papers in this direction. Now, what are these endonucleases? These endonucleases are called as homing endonucleases or meganucleases. They're called meganucleases because they are actually having very big recognition sites. Okay. Anyway, this book many of you may have read. So genes are in general referred to as being very selfish, and they try to inherit themselves while leaving behind their partners in the genome, and they will carry along with them only those partners which will be, helping them in their own survival. It's not just about inheritance of a large set of genes, it's about the inheritance of the gene itself. And for that reason, genes are known as selfish gene. But this particular homing endonuclease was found to be the most selfish gene in the genome ever known. So these endonucleases or meganucleases are highly specific DNA cleaving enzymes. That means they are DNA endonucleases. They are found within all forms of microbial life, as well as in eukaryotic mitochondria and chloroplasts. Remember, it was discovered in yeast. That are encoded by genes that display genetic mobility and persistence. So a little bit complex city arising over here. So see, these endonucleases are located inside introns, and they can mobilize themselves to other places, other similar places. So we'll show you those pictures very soon. They have a large recognition site. So endonucleases have recognition sites we have discussed all these days. Because the recognition site is very large, they occur rarely in the genome. So you know, as for a 4-cutter, you have more likelihood of finding the sequence than a 6-cutter. And if this sequence become bigger, finding that sequence in the genome become rarer. So it sometimes happens, these endonucleases may have only one single restriction site in the entire genome of a particular organism. Now these endonucleases are produced by genes which we call as homing endonuclease gene. So homing endonucleases, very simple, are proteins. They are endonucleases, they are enzymes, and they are produced by genes. And these genes we call as HEG. Okay, and you can see here in this top figure that these homing endonuclease gene is lying inside an intron or an intine, right? It may be inside an intron or inside an intine. So intine means that sometimes get included in the protein. Okay, we have cleared the concept of the intine earlier. Now what happens for these homing endonuclease, the target sites are on homologous chromosomes, which will not have the HEG gene, but it will have some sequence like this HE homing endonuclease target. You can see in the orange color over there. Okay, so this HEG gene product will come and cleave this particular ACS target and mobilize itself. The information will be mobilized here. That's why we will call this as a hosting gene, right? So let us take a small break for 5 minutes or 2 minutes. Okay, is the video coming? Fine. Okay, so how many participants are there? 86. Okay, thank you. So you keep some record of the attendance also in between, right? Just screenshot there are 36 people. Sorry. So let us now try to know more about these homing endonucleases, which are double-stranded DNases, and you already know they have large asymmetric recognition sites, and you will be amazed to know these recognition sites are not 4 base pairs or 6 base pairs. It may range from 12 to 40 base pairs, and you can calculate out the probability of finding a 12 and a 40 base pair sequence in the entire genome. So it may turn out that this particular sequence may be available just once in a particular genome. So that way, these homing endonucleases may not be that harmful, okay, but still they play a lot of critical roles in the evolution. And then when you think about these endonucleases from the point of view of genetic engineering, right, it may be very difficult to find out sites of these such big size to which these can be deployed. Okay, so anyway, so they are extremely rare sites to find. Okay, but some of the good things about this enzyme is that unlike the restriction enzymes, homing endonucleases tolerate some sequence degeneracy, right? So they may not exactly require the exact sequence. A little bit of degeneracy they can cope up with, and they will still carry out the work. So okay, so a few changes at the base level do not abolish the cleavage, but reduces its efficiency to some extent. Now, how does these homing endonuclease genes move to their cognate partners or to their homologous partners? This process is known as homing because it is not going anywhere. It is coming to its home only. But something like that, you are a single person. You have a home in one city and you have a home in the village. So you can stay only in one place, not both the places at the same time, right? So when you are staying in the village, you go to the city and stay there for some time or years, maybe for some particular work or job or study or whatever. And once everything is done, you may decide to go back to your countryside house or to village house, right? So for you, it is a homecoming. You are going back to your home. And after staying for a few years, months or week or days in your village home, you again travel back to your city home or your town home, right? You are actually not going to a stranger's house or somewhere else. You are again going back to your home. So I think this analogy of your, many of you may be having a home in the village, a home in the city, or you may have home in two different cities, or you may have home in two different villages, right? So you keep moving between these two homes. So similarly, these endonuclease genes, which are found inside introns, which is a very unusual fact discovered at that particular point of time, they keep moving between their homologous partners. But how do they do it? You go to home maybe by some transport mechanism using your own vehicle or by public transport, right? Or if it is nearby, you just walk. But in this case, we are trying to understand how these homing endonuclease genes go to their homes, right? So these homing endonuclease genes encode the protein. Somebody is switching on the microphone. Yeah, some disturbance coming. So I mean, I don't know how it is possible. Kindly please switch off the microphone. Just check with. Just check with. Huh? Huh? What's the name? Suprita, please kindly. Yeah, Suprita, please kindly switch off your microphone. Anyway, so they encode a protein in heterozygous cells and recognize the sequence of the homologous chromosome and cleaves it. The HEG is located in the middle of the recognition sequence, and so the chromosome carrying the HE is protected from being cut. After cleavage, the cell's broken chromosome repair system is stimulated, and the system uses the HEG containing chromosome as a template for repair. As a consequence, the heterozygote is converted to a homozygote. Okay, so you can see here this homing mechanism, and if you are interested for details, kindly read this paper by Austin, the book Genes in Conflict: The Biology of Synthesis Genetic Elements. Okay, so this is to show you in detail how homing endonuclease induces double-strand breaks and causes homing. You can see the blue pigment colored symbol is the homing endonuclease gene, okay, which produces this enzyme. Okay, the DNA ends of the cleaved recipient, shown in red, are engaged in double-strand break repair. Double-strand break repair has been already discussed earlier many times by Professor Tamuli and also Professor Celesting. Okay, so you understand this gene conversion in this case now. Now there are some generalized homing mechanisms for mobile group 1 introns and inteins. So here you see on the left side the group 1 introns, okay, and on the right side you see the inteins, and you can see the HEZ gene located in the center of this intron and intine. So when they are transcribed in the mRNA and the pre-mRNA, you have the HEZ information included. But in the case of left side, you see there's a splicing, and in the splice variant, you do not have the HEZ included or the intron included, and it is separated out. Okay, but it may happen in certain cases the HEZ may get translated. Okay, and then that may be used for cleaving the host to which it want to mobilize itself. On the other hand, you can see the intine is getting translated, and then in the process of splicing, the extins are removed or they are separated out, and this homing endonuclease gene is produced. Okay, so here in step 1, you can see the activity of endonuclease, which is translated either as a freestanding protein from the intron or as a fusion with the surrounding intine. On the right side, you have this protein with the red flanks. This green protein is having some of the information from the surrounding intine. So both of them, either in this free form or fusion form, they will make cleavage. This is a double strand break cleavage, and this will drive the homology driven repair. So this is in the second step. Subsequent repair via homology driven strand invasion and recombination and DNA replication using the allele containing the intron or intine. This completes the homing process. So this is how the HEZs home themselves. So this homing mechanism of mobile group 1 introns requires the translation of the intron encoded nuclease or the intine encoded nuclease. This protein product is highly specific for the intronless allele, where the intron is not there, binding to a homing site composed of flanking exon sequences. Once bound to the homing site, the endonuclease cleaves this site and cellular mechanisms, relying on homologous recombination between alleles for proper double-strand break repair, because the lateral transfer of the intervening sequences to the cleaved allele. So similarly, you have separate mechanisms for group 2 intron. Here you can see the transcription of gene X with intron, and then in step B you can see the translation of internal ORF introns or entire gene, the intine. Okay, and in group and step C you see the endonuclease or endonuclease containing RNP, the group 2 introns, binds and cleaves homing site on the gene X, lacking intervening sequence, and finally cellular double-strand break repair mechanisms take place as usual. So the homing mechanism employed by mobile group 2 introns are termed as retrohoming, and it differs significantly from the mechanism used by group 1 and archaeal introns and intines. As determined from group 2 introns found in yeast mitochondria and the bacterium Lactococcus lactis, this mechanism is more complex and requires the use of a ribonucleoprotein consisting of the encoded protein endonuclease and the spliced intron RNA. The protein also functions as a maturase and reverse transcriptase. Once the intron encoded ORF has been translated, the protein product binds the RNA and H in proper splicing. Okay, so let me cut short and just tell you that the homing mechanism of group 1 intron and group 2 introns are not same, they are different. And since these are endonucleases and they cut, DNA sequences, so we can use them for gene targeting. And this gene targeting follows the usual, double-strand break repair mechanism or non-homologous end joining inactivation process. Okay, so these are very, very common themes you will find in all kinds of genome editing technologies. Now, if you just compare the homing endonucleases with their relatives, the restriction enzymes, so in terms of conserved protein motifs, recognition sequences, accessory molecules, and genomic location, you'll find they are different. Okay, so these endonucleases, the restriction enzymes as a whole class do not have any conserved protein motifs. Okay, and but these homing endonucleases has 4 types of conserved protein motifs. You can see there Lugly, their G and GIY and YIG, then HNH and his cis. The recognition sequences already we told you are very, very long here, and they are asymmetric in case of homing endonuclease, but they're symmetric, and they may be asymmetric in case of restriction enzymes. They are sequence tolerant in case of endonucleases, homing endonucleases, but restriction site, restriction enzymes, no, they are very, very sequence specific. So some require protein or RNA components. We just tell you about the requirement of a ribonucleoprotein in the earlier case. In the case of restriction enzyme, they may require methyl transferases or specificity subunits. The genomic location of the homing endonucleases we discussed in length up till now. They may be in the introns, intine, or intragenic. They're found in all 3 biological kingdoms. Whereas in restriction enzymes, the flanking modification gene, and then confined to archaea, bacteria, and some eukaryotic viruses only. Now, these are the 4 different conserved protein motifs. So you can see that they have various members. The first one is the omega ISCE1, that's the Saccharomyces cerevisiae. It is a monomer. This was the first homing endonuclease to be discovered, right? And it was natural function was in the mating, okay? And that was the point from where it was discovered, right? And you can see the second motif, which is a modular DNA binding and catalytic domains it has found from the polyphases, okay? Then you have the other families, this PDDK, XK, exists as tetramers. So their notable features are motif also in recombinases, resolvases, and DNA repair enzymes, and so on. So this is in brief. I would like to actually finish it in the next 10 minutes because today we are keeping a small practical for you because that will help you in skilling up. Some of you may be already knowing some of these things, but anyway, for others, it will be very, very important. So I won't go into details of this homing endonucleases family. They are grouped into 5 families based on the sequence and small conserved amino acid structural motifs. All those motifs we just discussed over there. And some of these motifs probably will be also discussed in the practical that we are going to do. So this lecture material we will send you where you can read that these LHE genes exist in a variety of genomic environments and they are encoded within group 1 introns found in archaeal RNA introns as free standing units and so on. So you can see these homodimers viewed down the saddle group, which is proposed to accommodate the DNA. So you can see this beautiful structure over here, and you see a saddle over here. So you imagine the DNA double helix. This saddle sits on the DNA double helix, okay? And then if you can see my image over here like this, right? So this saddle is sitting over here, and then it will move around, okay? And this is the view from the perpendicular side to the saddle groove. So these are two different views of this homodiverse. Okay, so let me actually go straight away to the general properties of these homing endonucleases. Homing endonucleases are small proteins, around 300 amino acids. Okay, 235 was predicted earlier, but then when more and more of them were found out, they found out, okay, there are a lot of variations found in bacteria, archaea, and in unicellular eukaryotes. A distinguishing characteristics of homing endonuclease is that they recognize relatively long sequences. You already know, 12 to 40 or 14 to 40, compared to other site-specific endonucleases like restriction enzymes. The lengthy recognition sites and the name of the first known enzyme, omega, have given rise to the term meganucleases. So we call them meganucleases because of this particular region. We know that they do not have much sequence specificity. And at structural level, there is great disparity between the number of contacts made by restriction endonuclease and homing endonucleases. Restriction endonucleases exploit most of the potential hydrogen bonds between the proteins and their target sites, whereas homing endonucleases utilize only a fraction of the possible hydrogen bonds. So there is a difference there, and there is a lot of tolerance by the homing endonucleases. So despite the imperfect fidelity, the lengthy recognition sites can make homing endonucleases highly specific, often cutting large genomes only once. This attribute makes the homing endonucleases amenable to genome editing, where spurious off-site cleavages are detrimental. Okay. The DNA mining and recognition of homing nucleases, I will not discuss this in length. We discussed a lot about nucleic acid binding proteins. This is also one class of those nucleic acid binding proteins. So you now know a lot about the various elements involved, right? Whether it is the major groove or the minor groove or the shape readability, sequence readability. So let us not discuss those today again. But of course, there are certain things which are specific to the homing endonucleases. Let us go to one thing which is a little bit different over here. How do you name the homing endonucleases? Okay, for example, I am giving you here ICRE1. Okay, so this is a homing endonuclease whose gene was first discovered in the chloroplast genome, okay, of Chlamydomonas reinhardti, a species of unicellular green algae. So how is this name coming? It is named for the fact that it resides in an intron. The first I before CRE indicates that this is a gene product or a protein which is located in an intron. This is an endonuclease, but it is not the restriction endonuclease we found in the usual way. This is located in an intron, and it was isolated from Chlamydomonas reinhardti. It was the first. If you find the second member, it will be 2. So the remaining things are same as naming the restriction enzymes. Okay. So you put 3 letters similar to the restriction enzymes, but here the first letter is something important. I is coming from the intron, and if these particular protein contains intine, if it is an intine endonuclease, it will be written as PI. Okay, so ISCE1 and PISCE1 are not the same enzymes; they're different enzymes. ISCE1 is the mitochondrial large RNA intron omega endonuclease, and PISCE1 is a nuclear vacuolar ATPase intine endonuclease. Okay, let us be very, very clear about it. And F stands for freestanding, where the endonuclease is not intron or intin encoded. So it was proposed by Belfort and Roberts. This is another class. So we are unable to discuss at length about this at this moment. So here you can see a list of such enzymes. You can see the names on the left: ANI1, ICU1, ICHO1, I, and you can go below and you can see PIPSP1, PISCE1. Okay, so you have PISCE1 and you have ISCE1. They're different. Okay, so then you have below FSC2 and so on. So these are, F stands for the freestanding, PI for the intin, I from the intron. And these three-letter words, is coming from the first letter word from the, organism, and second from the genus and the species. Okay, that part is similar to the naming of the restriction endonucleases. Now, a lot of work has been done in creating highly specific nucleases by fusion of active restriction endonuclease and catalytically inactive forming endonucleases. So using these naturally occurring enzymes, some synthetic enzymes have been developed, and they are used for genome editing. So some of the important concepts are already discussed. They are double-stranded DNases, target large recognition sites, promote efficient gene targeting through double-strand break-induced homologous recombinations, and so on. And then finally, I would like to thank you. So now the next part of the class will be conducted by Adiraj Nath, who will do some practicals. Thank you.